Good afternoon, everyone. Welcome. I'm Clayton Rose. I'm the president of the college. I would like to welcome, in particular, our families who are back for Family Weekend. It is awesome to have you here. Today's Common Hour is the 2017 Arnold D. Cates Lecture, and it's sponsored by the Arnold D. Cates Lecture Fund. The fund was established in 2000 by Mark Garnick, Bowdoin class of 1968, and his wife Bobby Cates Garnick, in honor of Bobby's father. The original purpose was to celebrate creativity and innovation in health-related fields and to share that information broadly. And the Garnicks more recently have expanded the terms of the fund to include other areas of study. And going forward, the Cates Lecture will become a regular part annually of Family Weekend. The program that you've received provides information about Mr. Cates and the reasons that he inspired the fund, as well as about Mark and Bobby. And I encourage you to read about each of these accomplished members of the Bowdoin family. We are very fortunate to have Bobby and Mark with us here today, and I invite you to join me in recognizing them for their great generosity to the college. Thank you. I am delighted to be introducing our guest speaker this afternoon, Erin O'Shea. Erin is president of the Howard Hughes Medical Institute, as well as the Paul C. Mangelsdorf Professor of Molecular and Cellular Biology and of Chemistry and Chemical Biology at Harvard University. I can't do that all in one breath. Erin is also a friend and a colleague. I've been fortunate enough to serve as a trustee of the Howard Hughes Medical Institute for a number of years and have gotten to know Erin very well. In addition to being one of the nation's most accomplished scientists, she is a visionary leader and is leading HHMI into some exciting new areas. And the Institute, for those of you that don't know it, is the nation's largest private funder of biomedical research and an organization that is at the center of setting the agenda for basic scientific research in this country. Erin was named an HHMI investigator in 2000. She was elected to the National Academy of Sciences in 2004, and both of these speak to her position as one of a handful of the leading scientists in our country today. She became HHMI's chief scientific officer in 2013, and in 2016 was elected president of the Institute. She earned her PhD in chemistry from MIT and a bachelor's degree in biochemistry from Smith College, another great liberal arts college. Too bad it wasn't in Midcoast, Maine, Erin, but we're not all perfect. Erin is regarded as a leader in the field of gene regulation, signal transduction, and systems biology, and her research focuses on the way that cells sense change and adapt to that change, and it has deep implications for, among other things, cancer and other related diseases. In her talk today, How Cells Keep Time, the Cyanobacterial Circadian Clock, Erin will help us understand how it is that cells actually do keep time, and more generally, how it is that scientists think about the process of discovery and research. It is my pleasure to welcome Erin O'Shea to Bowdoin College.
Okay, thank you, Clayton, for that wonderful introduction, and also really for this opportunity to come here. I've had a terrific day and a half so far, and I'm really delighted to have this opportunity to share with you some of the work from my lab. And although the topic may sound a little baroque, as Clayton alluded to, I've selected this topic in the spirit of this day and the poster symposium that will follow, in which students will present their research. I thought, given that context, it would be appropriate for me to talk about some of the work from my lab, how we select questions to address in biology, and then how we try to answer them. And I'm going to tell you today about what I think is really just a remarkable aspect of biology, and that is the fact that within almost all of the cells in our body and in the bodies of many other organisms reside ways of keeping track of time. And so those are clocks, if you will, inside of cells. They're called circadian, as you'll see, because they keep track of time with 24-hour periodicity, meaning they repeat every 24 hours. And so this really is a remarkable thing when you think about it, right? That we have ways of keeping track of time inside of individual cells. And so I want to tell you a bit about what we've learned about how cells keep track of time and why this might be generally interesting. Okay, so as I said, circadian clocks are 24-hour biological oscillators. So all this means, as I said, is that they are clocks that keep track of time with 24-hour periodicity. So this wristwatch here, which has 12 numbers on it, keeps track of time with essentially 12-hour periodicity, right? So if I pick any point on this wristwatch, it takes 12 hours to get back to this same point. And that's what I mean by periodicity, and we'll talk about this on the next slide. So these clocks keep track of time with 24-hour periodicity. So if I start at a given point, it takes 24 hours to get back to that same point. These clocks are critical for controlling a number of aspects of physiology inside organisms. Organisms like humans, but also organisms like the fruit fly shown there, which we'll come back to in a minute, plants, mice, and also some bacteria, which is what I will talk to you about today. So with respect to humans, the clock controls a number of important physiological processes, things like when we sleep and when we're awake, how alert we are, and when we eat. And you all know this because you know that when you fly to a different time zone, you feel jet lag. And as we'll talk about, this jet lag is an issue with the way in which your clock, the lack of synchronization of your clock with the change in the environment. So a very special property of all real circadian clocks is that they keep track of time even in the absence of any environmental cues. So this is opposed to a situation where you could imagine like an hourglass that required the night to be reset in order for the hourglass to be turned over each day. That is not what a true circadian clock is like. A true circadian clock is like this watch here. The time is running independent of what happens in the environment, independent of whether it's dark or light out, for example. So organisms of all different types, as I've said, have evolved these clocks over a long period of time to allow the organism to take advantage of the predictable changes that occur in the environment, right? The sun comes up every morning and sets every night. And many organisms exploit that predictability to schedule various physiological processes like sleep to occur at the right time. You want to sleep during the nighttime and be awake during the day. And so the clock has evolved to exploit the predictability in the environment in order to be able to schedule various biological processes. So as I said, the hallmark of circadian clocks is this periodic output, and that's what's depicted schematically here, just output from the clock as a function of time. And as I told you, the clock has 24-hour periodicity, and that means if we start at a given point, it takes 24 hours to get back to this same point, and then the cycle repeats all over again indefinitely. So that's what we mean by the period. The period of the clock is 24 hours. It's the time it takes to repeat. The other term I need to introduce is phase. And so the phase is the relative relationship between two of these oscillators, between two of these clocks. And you should all care about this because this is what happens in jet lag, right? Your clock is oscillating with a phase that is not synchronized with the environment. It's not synchronized with the day-night cycle when you go to a different time zone. And your clock must adjust. It must become synchronized with the new environment in order for you to get over jet lag. And so this lack of synchronicity is a difference in phase, lack of synchrony. All right, so you've probably heard that the Nobel Prize this year in medicine was given for the discovery of genes involved in circadian clocks and for an understanding of how these genes work together to help keep track of time. And that prize was given to Jeffrey Hall and Michael Rosbash, as well as Michael Young. And I'll just summarize on the next slide sort of what their real discovery was, their conceptual discovery about how the clock worked. So they studied the fruit fly, Drosophila melanogaster, and they identified genes, right, that produce proteins. Those are the workhorses inside the cell, and those proteins were parts of the clock. And the way in which they, and we currently think that the clock in flies and also perhaps in humans works is diagrammed on this slide. So here, this blue bar here represents a gene. It's a piece of DNA, and this piece of DNA, of course, eventually will go on to produce, through a number of biological processes that we need not concern ourselves with, the gene will go on to encode a protein. So a protein will be produced from this gene through an intermediate that we call messenger RNA, and via processes that we refer to as transcription and translation. And I mention these only because the model uses these terms in the name. But you need not worry, just the information in this gene will produce this protein. And it is a protein involved in the clock. And what they postulated, Rosbash, Hall, and Young, is that this protein would be produced, and it would take time to accumulate inside the cell, right? And to be modified, to be changed to make it active. So this takes time, and so if we just plot the amount of this protein as a function of time, this schematic here shows that it just accumulates over time, all right? And when it reaches a sufficient level, what Rosbash, Hall, and Young showed and proposed in this model is that this protein then goes on to turn off its own production, right? And if it turns off its own production, then the levels of this protein drop because the protein X is no longer being produced. And now, so now you have part of the cycle, right? One full cycle, but it has to repeat again. And the way it repeats again is that this protein X, which is turning off its own production, is then degraded. It's removed. Okay? And that allows this cycle to start again. And so the clock in humans and in fruit flies and in many other organisms is thought to rely on this structure that's called transcription-translation oscillator. Right? And it's called that, as I said, because transcription and translation of the gene are required to produce a protein that then goes on to regulate its own production. Okay? So transcription-translation oscillators. And for this, as I said, they were awarded the Nobel Prize. All right. So what I want to talk to you about is how we've unraveled the way in which the clock inside a different organism works and how it's similar and different from this picture that I just showed you on the previous slide. So the big questions I want to talk about are, what are the parts of the clock? And I want to talk about how we figure that out. How do we know what are the parts of the clock? And by parts of the clock, I mean, like if you open up a wristwatch, you see a complicated set of pieces. How do we know which of these pieces are critical for keeping track of time? We also want to know, how is information about time read out by the cell, right? How does the cell know what time it is from this clock that's operating inside it, right? With a traditional clock or my wristwatch, I know time is read out by the position of the hands on this clock. But there are no hands inside the cell that are representing time. And so we want to know what the sort of molecular analog is of the hands on the watch. How is time read out? And as you'll see, this is a very elegant solution that the cell has to read out information about time from this clock that is contained within the cell. And finally, and perhaps the most, you'll see, mysterious, is how are oscillations generated, right? How do you generate these oscillations inside the cell that enable the cell to keep track of time? And I've shown you one way that was proposed by Hall, Rosbash, and Young. And you'll see that the organism we work on has found a very different and also very elegant way to generate oscillations. And I'll talk about how it's similar yet different from the solution the fruit fly has found. So the organism that we work on is a crazy one. They're called cyanobacteria. This is a picture, a microscopic picture of the cyanobacteria. They're single cells. They look green when you grow them in a flask. They live in water. Some live in fresh water, some live in the ocean. These organisms are interesting and have attracted a lot of attention because they rely on photosynthesis. They rely exclusively on photosynthesis to get energy. So photosynthesis, right, is what all kind of plants do. They use the energy derived from light, from the capture of photons, light, to produce a chemical that is the currency, energy currency inside cells. And that chemical is, of course, ATP, or adenosine triphosphate. So photosynthesis is this process, it's a very complicated process by which many different organisms, including this bacteria, but plants, can convert the energy inherent in light into a chemical that they can use for energy, this ATP. Cyanobacteria are the ancestors of chloroplasts. This is where all the photosynthesis takes place inside of plants. So chloroplasts came from cyanobacteria. They're responsible for the majority of photosynthesis in the open ocean, and in fact, a significant fraction of photosynthesis on this planet. Many cyanobacteria, in fact, almost all of them, have a circadian clock. And now, based on what I told you, you can imagine why this might be. So the cyanobacteria, they rely on photosynthesis. So this means they rely on sunlight to make energy. And so you can imagine why it might be advantageous for them to schedule the process of photosynthesis to take place only when light is available. You don't want to try to do photosynthesis at night because there are no photons, no lights available. So many of the cyanobacteria, as a result, have evolved this circadian clock. And we study them because we can do a lot of experiments in this organism that you can't do even in the fruit fly. They're extremely easy to work with, and we can do a lot of genetics, as you'll see. So one of the sort of coolest things about the cyanobacteria is that we can see, and I'm going to show you, we can really see circadian rhythms. We can see the functioning clock inside of single cells. And we can watch what happens to the clock, how it changes over time. And the way in which we do this is using this thing we call a transcriptional reporter. Okay? The term here doesn't really matter, but I might say it, so I'm pointing it out. And all this is a gene, so a piece of DNA, that will produce a protein, right, the workhorse inside the cell, that can produce light. When you shine light of a given wavelength on this protein, it will produce, it will emit light of a different wavelength. Okay, that's fluorescence. Absorb light of one wavelength, emit light of a different wavelength. Okay, and so we can measure the emitted light, that's fluorescence. So you can just think of this as like a light bulb inside the cell. And it turns out it's reporting on the clock. And the reason it's reporting on the clock is that there is a piece of DNA that controls the transcription or expression or production of this fluorescent protein. And that control element, which is a piece of DNA, is responsive to the clock. So this is an indirect way of looking at the output from the clock, right? So this black thing controls the expression of the green protein, that's the light bulb, and it's responsive to the clock. And so if we measure fluorescence as a function of time, you see this is a schematic, but what we see is that the fluorescence oscillates with 24-hour periodicity, right? It repeats every 24 hours, and this reflects the activity of the clock. Okay, so as I said, you can really see this. And so this is a movie. And on the top, you see these dark little rods. Those are the cells. And on the bottom here is the fluorescence image. This is, now you're looking at the light bulb, essentially inside these cells. And I'm going to start the video, and you'll see the cells grow and divide. They get longer, and then they divide in half, et cetera, et cetera. But pay attention to what happens to the light coming from the light bulb, the fluorescence here. And what... Oh, no. Yeah. What you can see it gets bright and then dim, and then bright and then dim, and then bright again. Okay, so really, it's amazing. I mean, we are looking indirectly at the clock, and you see a few things. I mean, one is that you can't tell that it oscillates with 24-hour periodicity, but you have to take my word for it. It does. And the second thing is, and we can come back to this in the questions, you see all the cells are basically marching in lockstep. They're all getting dimmer and brighter and dimmer and brighter, more or less at the same time. Okay, and so that's another interesting aspect. But so we can use this tool to study the clock. And one thing we can do with the tool is to ask, what are the parts of the clock? And we know what the wristwatch looks like. If we open it up, there are a whole bunch of springs and wheels, and each of them has a specific function. And now this isn't complicated to know which parts of the clock are important. I'm sure your intuition would tell you the experiment that you would do is to take pieces out of this wristwatch and ask what happens to its ability to keep track of time, right? And some of them would have more of an effect than others. And that would allow you to conclude that those pieces that you pull out when the clock is broken, that tells you those pieces play an important role. And that's exactly what we do. And this was work done before I got involved in this project. Kondo, a Japanese lab, now about the same time as the work was done in Drosophila, for which the Nobel Prize was awarded, Kondo looked for cyanobacterial cells that no longer show oscillations, right? So the normal cells show this oscillation that I showed you that we can measure with this reporter. And he looked for mutants. That just means like defective cells that don't oscillate anymore. And then he identified the genes, the pieces of DNA that contain the mutations, that contain the changes that broke the clock. Right? And then he studied the proteins encoded by those genes. And this is also where my lab came in. And it turns out there are 3 clock proteins. They're called Kaic, Kib, and Kaic, and I've just drawn them schematically here. And it turns out they comprise the clock, only these 3, in the cyanobacteria. And so now I have to tell you just a little bit about these proteins. And the most important of them is this protein called Kaic. So Kaic is the only one that really has activity that acts as an enzyme, meaning it can really make a chemical modification. And what it does is to add this group here. This is a phosphate. It has phosphorus, the P, and oxygen bonded to each other. And then these two minuses represent negative charges. Okay? And this phosphate comes from ATP, this energy currency inside the cell that I was talking about. And what happens is Kaic can put phosphates, attach them to itself. And so that's what these little black balls represent, the attachment of this phosphate group onto the Kaic protein. All right, this process is called phosphorylation, but that doesn't really matter. I'll talk about the black balls being put on Kaic. And we can measure inside the cells the amount of Kaic that has the black balls on it, okay? And it oscillates with 24-hour periodicity. Okay, so now we're measuring the clock directly, and we see this 24-hour periodicity. So Kaic is phosphorylated in vivo with this rhythmic periodicity. And it turns out that Kaic has two activities. It can both put the black balls on and it can take them off. And for those of you in the know, the putting them on is kinase activity and the taking them off is phosphatase. But I'll just refer to them as putting the black balls on and removing the black balls. And the black balls are important. They change the activity of this protein, okay? That then affects output from the clock, affects the ability of the clock to control physiology, to control photosynthesis, okay? So the black balls are important. And the other thing I should say, and now here was the first huge surprise, and there are going to be two big surprises here in the first part of this talk, and that is that the oscillation doesn't require transcription or translation. So remember I told you, Hall, Rosbash, and Young won the Nobel Prize this year for the discovery of this transcription-translation oscillator in Drosophila. And that means oscillations and the clock function rely on these processes. But this clock doesn't. We'll get back to this, why this is different but yet similar, towards the end if I have time. So the most remarkable thing about this cyanobacterial clock is the following. And that is that you can take these three parts of the clock, Kaic, Kib, and Kaic, they're proteins, and mix them together in a test tube with ATP, this energy currency, and an ion called magnesium. And you can reproduce oscillations in the amount of Kaic that has black balls on it. Okay, so this is really amazing, right? Taken out of the cell, the proteins alone make oscillations and keep track of time. This keeps track of time indefinitely as long as you supply ATP. Okay, so that's a huge mystery, right? And we're going to talk about how this happens, right? So only 3 purified proteins are needed to keep track of time in this organism. And it turns out, the other thing I need to tell you about this interconversion of the Kaic that has no black balls and the Kaic that has the black balls is that that's the role of these other 2 proteins. So when this protein Kia is present, Kaic puts black balls on itself. Okay? So in the presence of Kia, we get this Kaic with black balls. And in the presence of Kib, what Kib does is to inactivate Kia. So if Kia is inactive, the black balls get removed by Kaic. All right, so with Kia, we get black balls. With Kib, we get no black balls. All right. So now you see a lot of you see how oscillations might come about, and I'll tell you how this works in a minute. And you see the role for these two proteins, Chi A and Chi B. They control the activity of Chi C, meaning they control the ability of Chi C to put these black balls on itself and take them off. All right. So now you know about the parts of the clock and this remarkable ability of the clock to keep track of time in a test tube with just 3 pieces. Now we want to understand, we want to use that system to understand how is time read out, right? So in the wristwatch or this clock here, it's read out with these hands. And I told you about some ways to read out time in this system, right? And I specifically have said, I told you about the reporter, but closer to the clock, I told you about reading out the amount of Chi C with black balls, right? That oscillates with 24-hour periodicity, and that it's an important part of the clock. But this can't be how time is read out. And the reason is, just like this wristwatch here that has 12 hours on it, when I look at 3 o'clock, I don't know whether it's AM or PM. And there's the same problem here, right? If I read out time by reading out the fraction of Chi C that has black balls on it, I could never know whether I'm in this rising phase of the oscillation or in the declining phase. So the oscillation passes through this same point, the same amount of black ball Chi C in both phases. Okay, so that can't be how time is read out. And it turns out the answer about how time is read out is really, I think, very elegant and simple to understand. And it lies in the details of those black balls that I told you about. So it turns out that there aren't just black balls. There are 2 kinds of black balls, red ones and green ones. No, seriously, this is easy to understand. Now you can put red balls on Chi C or green balls or both. And now you can imagine what the answer is here. So there are 4 possible kinds of Chi C. It has no balls on it, red balls only, green balls only, or both red and green balls. Okay, and it turns out what we did is to measure the amounts of these forms of Chi C as a function of time, and what we found is something very cool. So here in black is just black ball Chi C, not looking at the individual green and red balls. And now here on the bottom are green ball only, red ball only, and blue represents molecules that have green and red balls. And you see that all of these different forms of Chi C, they oscillate with 24-hour periodicity. But the phase is different. Now remember the phase, they're offset. And now here's the secret to how time is read out. So what happens, I've already said that, each form oscillates with a different phase. And time is stored in the relative amounts of the green ball Chi C, green and red ball Chi C, and red ball Chi C. And you can see this. So if we just compare these 2 points that have appeared to have the same level of black ball Chi C, when we look in depth at and break down the modification into green, red, and blue, you see at this point there's a predominance of the green one and then equal amounts of blue, which has both green and red balls. And over here, the red one predominates. So the cell can tell what time it is by monitoring the different amounts of Chi C that's modified with these green balls, red balls, or both balls. That's how time is read out. And now we want to know, how are oscillations generated? And in the wristwatch, a mechanical wristwatch, it's actually complicated. There are all kinds of pieces, a mainspring that stores mechanical energy, and a balance wheel that's a weighted wheel rotating back and forth that's controlled by that spring, and that sets the period. I mean, this is also a fascinating issue, right? What sets the period? And I think we understand this with the wristwatch, and you can ask me in the questions how the period is set in the circadian clock. That's a whole other fascinating topic. But we want to know, how do you get oscillations, right? I've told you about the 3 proteins and how the time's read out in the different balls on Kaic, but there's a huge issue about how you get stable oscillations. I told you about these 2 proteins that control whether Kaic puts black balls on itself or removes them. And if you think about this, there's a big question about why the reaction doesn't just go to what we call a steady state. And that's just some fancy word that means not changing over time. And I think I can make a good analogy here. If one has a bathtub where you're putting water in and draining water out, the level of water that you have, if you're both putting it in and draining it out, is determined by how fast you put it in and how fast you drain it out. And eventually, if you turn on the faucet and you open the drain a little, the water will reach some stable level that's determined by the relative rates of water in and water out. And you know this from your own experience, and that is this steady state. So this is the same kind of setup here. 2 opposing reactions, water in and water out. And the relative amount of black ball Kaic should just be determined by how fast the balls are put on and how fast they're taken off, just like the water in the bathtub. And so what we see instead of this steady state are these oscillations. And this would be as if you put water in the bathtub, you turn it on, and you open the drain a little, and the water oscillates up and down. Okay, and that is not what happens, but it is what happens here. So this is really, I mean, it turns out it's a remarkable thing, and there are very few systems in biology that exhibit this property. And I'm just going to tell you that what we need is some time delay between putting the black balls on and taking them off. If they're put on and taken off with the same time scale, you can't get oscillations. And I think this will become clear why in a minute. And the other bigger problem is that there are billions and billions of molecules of this Kaic protein and the other KaiA and KaiB proteins in a test tube. And just like the cells I showed you, this is an even harder problem. To get oscillations, these molecules need to be marching in lockstep. They have to be putting the black balls on and taking them off, almost all the molecules at the same time. They can't be asynchronized, right? They can't be out of sync, if you will. And that is a hard task. How do you keep the billions of molecules in sync with one another? And that's this synchronization. And you'll see the answer lies in those 2 proteins, KaiA and KaiB. And so we figured this out by studying in a test tube these subsets of clock proteins. So Kaic with no black balls all by itself, what does it do? Kaic with this KaiA proteins that tells it to put black balls on, and then Kaic with Kaib. And I'm not going to show you any experiments. I'm just going to tell you what we learned. Yeah, exactly. So putting the black balls on and taking the black balls off of Kaic are really slow. And this is like crazy in biology. Normally in chemistry, reactions normally take place on a time scale that's seconds or less, very fast. But here, these reactions are really slow. And this is just a property of this protein Kaic, and we can talk about why at the end, but this is what gives rise to the period being so long, okay? And it's a peculiarity of this protein. So that's one thing we learned from these reactions. And the other thing we learned is that if we measure Kaic with the green balls, the red balls, and both balls, what we see is the balls are put on and taken off in a very stereotyped order. So what happens is the molecules essentially always put the green ball on first. Then they put the red ball on to make this blue thing. And now both balls are on, right? That's this, don't worry about the S and the T, but both balls are on. And now the protein always removes the green ball first to leave the red ball right there. And then the red ball is removed to make unmodified Kaic. Okay, so there's some order here. And it's the same order we see when all the proteins are present. And now we can see we get oscillations. To get oscillations, we need this delay between putting the balls on and taking them off. And the delay comes from this order. It is just a property of the enzyme, and it's obligate. It has to happen this way. You start with no balls on, put the green ball on, put the red ball on to make this both balls there. And now there's a delay. There's a delay in removing the balls. There's nowhere to go here except to remove balls. And it always removes the green ball first to leave the red ball, and then the red ball is removed to produce the unmodified form. So the delay comes from this order here in the cycle of addition and removal of the balls. Okay, and now I told you that we need some way to keep all the molecules marching in lockstep, right? And this is really a serious problem. And the answer turns out to be a very elegant solution. So here you're adding green balls, then you add the red ball to make both balls on there. And the Kaic protein I told you is telling Kaic to put the balls on. And now what happens, and the way the cycle is reversed, and the way the molecules are kept in sync is as follows. I told you next, after both balls are put on, the green one it's always put on first and removed first. And it's removed, then you just have the red ball. In this form of Kaic with the red ball plays a very important role. What it does is to bind, it forms a complex with this protein Kib. And I told you, Kib then inactivates Ka. If you inactivate Ka, now no more balls are put on. And instead, the balls all get removed. So if you have two balls on, you go to this. If you have the one ball, you go to the unphosphorylated or unmodified, and eventually down to here, and you start the cycle anew. So the way the molecules are kept in synchrony is this special form of Kaic with the red balls triggers this regulation that inactivates the protein that puts the balls on Kaic. So it's really a crazy situation where all the molecules are bathed in a sea of these regulatory proteins, all the Kaic molecules. And if you control the activity of the regulatory proteins, then you control the activity, the modification of Kaic. So I've told you the parts of the clock are these three proteins, and ATP is critical in this system, this energy. That's where the balls come from. They come from ATP. I've told you about how time is read out. It's read out by these different forms of Kaic. Forms meaning Kaic that has these different balls. They're phosphorylation, modification by phosphorylation, but you can think of them as different balls. And that's how time is read out, and how the clock knows whether it's in the rising phase or in the falling phase of the cycle. I told you about how oscillations are generated by this form of feedback, effectively, that controls the phosphorylation of Kaic. And now, in the less than 5 minutes, I want to turn back to this issue of, I set the whole thing up with the Nobel Prize and the transcription-translation oscillator. And I told you that transcription-translation oscillator is how the clock in humans is thought to work, and how the clock in the fly is thought to work. And now I told you something totally different about these bacteria, right? Like nothing related. But it turns out that that aspect of transcription, translation, feedback that I showed you, the details of which don't matter, also occurs in the cyanobacterial clock, okay? So there's something very interesting going on here. And by what I mean by this is, so the clock is composed, I told you, of these 3 proteins that generate oscillations to control output, and it turns out they control expression of genes, including these genes, the clock genes themselves. All right, so the clock controls the expression of clock genes, and that's what this arrow means. So the clock is controlling its own expression, just like I showed you for the flies and what is thought to happen for the humans. And so the issue is, why do these bacteria have this other layer of regulation that looks just like the mechanism underlying that creates oscillations in the fly system? Why would the bacteria have that when I just got done telling you that the oscillations come from this crazy mechanism with the balls on Kaic? Right? Why do the cells have this? And I'm just going to tell you that the answer is a very cool answer. So we broke this, we can break this, an experiment that I won't talk to you about. And what happens is very revealing. So this clock essentially has 2 gears. So here's the gear with the balls being put on Kaic, that's what I told you about. And the cyanobacterial clock also has this transcription-translation feedback thing that I told you about that's operating in the flies and human. So this is indeed the balls. They are required and sufficient for the oscillations. But then why do we bother with this? And now I'm going to show you the only real piece of data besides this video, but it's easy to understand. So here in the top, now we're measuring this fluorescence, the light bulb, in the individual cells of the bacteria. And I've traced, you can barely see, but see these thin lines represent individual cells. And you see that more or less they're marching in lockstep. They have different levels of brightness, and that's why some are way up here and some are down here, but they're marching in lockstep with the same phase. That's the normal situation. We call wild type, but it's just normal cells. And now when we break this feedback that I told you about, the transcription-translation feedback, so now the clock doesn't control its own expression. And this is what happens. Now you see the cells are all desynchronized. They're asynchronous. They can't keep track of time in synchrony. And so it turns out that this feedback is required for the clock to stay stably in sync, the cells to stay in sync with one another. And that turns out to be the function of the clock. And so I'll end by just commenting that I think it's not clear evolutionarily. This is likely a very ancient clock, this circadian clock. And it may be that this is the ancestor of all circadian clocks, and that the regulation I told you about, for which Hall, Ross, Bash, and Young were awarded the Nobel Prize, has then taken over as the mechanism underlying oscillations in more complex systems. And this has been lost, but retained for whatever reason in cyanobacteria. We don't really know, but there's something peculiar going on here. And I'll just end by thanking the people in my lab who did the work. Really, all of the work on clock mechanism was done by one postdoc, Mike Rust, who's now a faculty member at University of Chicago, actually, and Joe Markson, who was a graduate student with me. And I'll stop there, and I'd be happy to take any questions.
Hi, so I'm really interested in the way that you talked about these oscillators, and my question is, you talked about how different cells synchronize with each other, but you also talked at the beginning about the environment. So if they're nice 24-hour cycles, how, if you get jet lag, do you get re-synchronized to the environment if that changes?
No, and I really sort of skimmed over this. I didn't tell you the answer to that, and you're very perceptive to pick up on it. So the reason the cells are out of sync with one another really is tied to this getting over of jet lag. So we synchronize the cells with one another by, we can do this with special signals. Special meaning, it's true of humans too. There are only special cues that can adjust the phase of the clock. Those cues are things like light. That's why they tell you when you go to a different time zone to be out in the light. Food, not in this bacteria, but in humans, food is also a dominant cue for the clock, and that's why you should eat on the schedule of the new time zone. So those things are special cues that help adjust the phase of the clock. And in the laboratory, we can use light or darkness, a pulse, to adjust the phase of the clock. And so we apply a pulse to a population of cells that are not synchronized, and we synchronize them with that pulse of darkness. And then they start all marching in lockstep. And then in this experiment that it was at the end that I didn't describe well, what happens if you don't have that feedback where the clock is controlling its own production? They quickly lose synchrony, whereas the wild type, the normal cells, do not. And we can talk offline about why that is. We understand a great deal about why they lose synchrony under those situations. But the answer to your question is, yeah, there are these cues that can adjust the phase of the clock, special cues. That's right. And that's a process called entrainment.
Hi. A follow-up on that one.
Yeah.
I know in another type of microalgae, dinoflagellates, there's been work showing it's a blue light receptor that can synchronize cell cycle, and I've heard of putting a blue light behind your leg for jet lag. Have you looked at all at blue light receptors in cyanobacteria?
Cyanos.
Yeah.
No, we've not. To the best of my knowledge, I don't know that they have blue light receptors, but this is an interesting issue in cyanos. It is an interesting issue, and in humans too. To which wavelength is the clock sensitive for this process of entrainment or getting over jet lag? I'm sure people have done such experiments in humans. We've not done them, and to the best of my knowledge, they've not been done in cyanobacteria. I'm not sure, dinoflagellates, I don't know if they have a clock.
So yeah, so she tested blue versus red versus green, and it was for cell cycle.
Yeah, for
blue
cell
and
cycle,
yeah, which
yeah.
is that circadian? Or,
Yeah, it's not, I don't know.
I don't
know. I'm not sure that they actually have a clock.
Yeah.
But it's still, your question is an interesting one. Yeah.
Thank you.
Can I ask you why 24 hours? If
Oh,
the circadian
yeah.
clocks operate independent of environmental cues, why not 22 hours or 26?
Okay, so I misled, that's a great question, and I misled you a little. They're really not exactly 24 hours. That's right. No, it's absolutely true. It's not exactly 24 hours. And that differs depending on the organism. It's right around 24 hours though, but I mean like it's 23 or it's 24.7 or 25, but so that's why we say circadian. It's approximately 24. And I think the truth is the system can never have a period that's exactly perfect, because in the environment, the fraction of the day that is light changes with seasons, with latitude, right? So you always need entrainment to register the clock to a point in the day. Yeah. So you see what I'm saying. So I don't think these small differences in period, I don't think we understand well enough we don't understand where those come from. What I can tell you is that there are mutants in the clock, in the bacteria. This is an easy one to study. And if you create a situation, you don't even need mutants. If you create a situation where the environmental variation, the periodicity of environmental fluctuation, like light, dark, doesn't match the oscillations in the clock, the cells have a fitness defect. They don't grow as well as cells whose oscillations match those of the environment. Yeah. So the clock, and this makes sense for the cyanobacteria because they're growing only when light is available. And they have to use a huge amount of energy to produce all these proteins that are involved in photosynthesis. And so they schedule it to produce those proteins right before they're needed. If they produce them all the time, they would waste a lot of energy. Yeah. Yeah. Apparently, I got.
That's right. The hook.
I got the hook.
Erin, that was fabulous. Thank you.